Good afternoon, everyone. I, Dr. Shweta Dhonse, welcome you all for today's forenoon session. It is my pleasure to introduce our esteemed guest speaker for today's technical session, Dr. Chetan Rayakar. Dr. Rayakar is a seasoned expert in the field of artificial intelligence and neuroscience and has made significant contributions to the advancement of artificial intelligence. He brings with him a wealth of expertise and experience garnered through his illustrious academic and professional journey as a postdoctoral research associate at the prestigious MIT, USA, which is one of the topmost ranking universities in the world. He has been at the forefront of cutting edge research in neuroscience. With a PhD earned from the esteemed Indian Institute of Technology, Delhi, Dr. Rayakar has demonstrated his prowess and dedication in the realms of artificial intelligence and cognitive science. His doctoral research, focusing on brain inspired machine learning, deep learning, has garnered acclaim for its innovative approach and potential impact on shaping the future of intelligent systems. Dr. Rayakar's journey is characterized not only by his academic achievements but also by his practical contributions to the industry. He brings a wealth of experience from his affiliation with HCL, where he has applied his expertise to real world challenges, driving innovation and progress in the field of technology. We are privileged to have Dr. Rayakar grace us with his presence. His insights and his insights garnered from his rich academic background and practical experience are sure to enlighten and inspire us all. So I would like to extend a very warm welcome to Dr. Chetan Rayakar. We eagerly anticipate the wisdom and knowledge that you will share on the topic advancements in AI and neuroscience. Welcome, sir, and over to you for the session.
Thank you so much. It's really an honor to be called in my alma mater to talk about my research. And thank you so much, ma'am, for a very kind introduction. I hope I am audible. And
Yes,
also,
you
I
are. Yes, you are.
was a coordinator just if at all my internet or if my screen is frozen or something, because I can't really monitor the chat once I share my screen. So just drop me a WhatsApp message if something goes wrong so that I will know that uh, everything is
Oh, sure.
working. Okay.
Please go ahead.
Um, I'll just start sharing my screen. Can everybody see the screen?
Yes, it's visible.
Okay. Okay. Just a second. Okay, thank you so much again for inviting me for this talk. So I'm not very sure about the background of the audience. So what I did is I am just trying to keep my talk very high level. And if you have any doubts or if you want to go in much more details of it, so I will be happy to take any questions on this. So in today's topic, I will be discussing about some of the techniques in AI that is little bit of my contribution, and at the same time, what are the current research projects that I'm handling at uh, MIT. So if I look at my journey, so I did like BTech in electronics and telecommunication, and then worked as a software engineer at Cognizant. And also I did master's in electronics engineering, specialized in signal and image processing from YCCE, and did PhD at IIT Delhi. And then I'm currently at MIT, and I'm working on two major projects at MIT. One is like Project Prakash, which is funded by National Institute of Health, which is more in terms of a scientific aspect or exploring the basic scientific questions. And I will be talking some of the portions of Project Prakash in today's talk. And I am also part of Intelligence Advanced Research Project activity, which is like US Intelligence Agency's project, wherein we are trying to understand how face recognition is achieved at different distances or a degraded face image recognition is the topic of that project. So in today's talk, I'm trying to divide the talk into two parts. And based on the time, I will just try to cover as much thing as I can. So in the first talk, I will be talking about more sort of an artificial intelligence model, which is like how we come up with a character recognition model, which is not basically a model which is based on engineering settings, but we have taken some inspiration from the cognitive experiments. And in second part, I will be talking about development of electrophysiological correlates of face and non-face distinction in children who are blind, born blind, and later in life they got the sight. So let's start with the part one. So as I mentioned, like character recognition, when the first thing come into our mind is optical character recognition. So optical character recognition is a system wherein we generally scan the documents and then convert it into editable format. So in initial days, the OCR, we call it as like OCRs were dealing with the printed characters or the constraint shape. So it was very easier for us to convert it into the editable format. As we move ahead, then this printed shape turn into the unconstrained characters, which is handwritten characters. And later on, we also have artistic and noisy characters. So now, as we see from printed to the artistic, the complexity of the character structure is is increasing. That's why the techniques which are needed for identification of such character also got some improvement in terms of the techniques. So we initially started off with the handcrafted characters, and now we have like feature extraction to deep neural networks. So now the question is, if we look at this, all of these characters, humans can easily identify the character irrespective of any variation in character structure. And at the same time, when we talk about neural networks, we don't really understand what's happening inside it and how the accuracy is improving and what is exactly the system is learning. So there are two aspects. One is a human aspect and one is the machine learning aspect through the deep learning networks. So if you look at the human, how humans start their journey to see. So the babies are born with a blur vision. Their vision gets improved over a certain period of time, like within few weeks after birth, they get a clear vision. After that, they interact with the environment, and then the face perception is developed. Later on, they are introduced to the different characters that they learn to write, and then as we progress, we come across the handwritten characters, and by observing the hoardings and while interacting with the environment, we come across this artistic or ornamental characters. So based on this journey, we can clearly say that humans have vast visual experience. At the same time, characters are overlearned pattern because every day we come up or get up in the morning, just look at your WhatsApp or any messages, then reading is, you need, it's like everyday activity. And that's how we are coming across various samples of the characters in different forms. If you look at this deep neural network architecture, we actually need millions of samples to make a deep learning model train on so that we can get a best performance. But human learning is like advancing through different phases of life. So we can say like training is through the visual experience. Now, the interesting question is, what do deep learning architectures learn, and how do humans learn, and what humans learn, or is there any pattern between their learning? If we know these two patterns, then we can say like we can bridge the gap between machine and human learning. Now, this has motivated us to formulate a question like, can we just propose an intelligent character recognition model by understanding strategies used by humans at the same time the strategies used by deep models? So in this question, there are two aspects. One is the strategies used by humans, and another is strategies used by deep models. So let's look at these two aspects separately. When we say recognition, so the if we look at the neural correlates of recognition, so we see that when we look at any object, then the the information falls into your eye, and then it follows a path from the primary visual cortex to the inferior temporal cortex. So this is the section of a brain where I'm showing this is the occipital lobe, which is responsible for processing your um, simple structure or simple information. So you can see the simple cells, like the information passes from V1, V4, IT, and then prefrontal cortex. So as we move along these lines, we can see that the simple features are learned in the initial layers. And as we progress, the simple features are converted into the complex shapes. And ultimately, the object is recognized at inferior temporal cortex. Now, in order to understand how exactly this recognition is happening, we need to monitor the activity of thousands of neurons which are getting activated by looking at an image. Now, in order to record that activity, we need a very sophisticated, invasive technique. Like we need to put the electrodes inside your brain, which is not really possible. So, what is not possible, as in it's not very feasible. So, what is the best way to go about uh, talking about strategies of humans? So there is a motivation. If I ask you to just tell me what you see in this image or how many tiger faces you see, can anybody want to say uh, how many tiger faces they are able to see? I'm not sure whether these participants are allowed to speak during this talk. So here, if we, yep, then thank you so much for your responses. So if you look at this image carefully, what is happening here is when you say there are these many number of tiger faces in this image, what you are actually doing is you are looking at different regions in that image, and your brain is processing that information, and then you are coming with an answer. For example, you might have focused on this point that a circle that are shown as a circle, like point 1, 2, 3, 4. And likewise, we can say like we point our eyes at relevant locations. Now, once you point your eyes at that location, you process that information, and then you move to the different point. Now, these points are called as focus points or fixations, and this fast movement between these fixations is called as saccade. Now, as I told you, like based on your processing strategy or based on your eye movement, we can comment on something. So eye tracking seems to be a promising technique to understand visual attention, like where you are focusing your attention, and based on that, we are collecting those glimpses, and at each glimpse, humans are processing that information and then come up with a kind of a solution. So classically, eye tracking is the process measuring the point of gaze, and the fixation is like maintaining visual gaze at a particular location for a certain duration. Whereas saccade is called as fast and simultaneous movement of eye. I won't go much details of how we actually do the eye tracking, but I want to just show you one of the snippet from my previous experiments. If you closely observe here, the task was given like this text was shown to the participant and they were asked to just read it. And I was also checking their comprehension level. So if I say, if you observe this, so now these circles are the fixation points where you are focusing while you read the text. And here is another participant who is also reading the same text. Can anybody tell me, or in the chat, you can say like what you can infer from those two patterns? Can you say something about this? What exactly is happening here in left and right eye gaze patterns? So what I'm trying to say here is these eye movements are actually getting you understand what is a language comprehension, how much language expertise that person is having. Because if you look at the right panel, the video in the right, I hope the video is getting played. That you see there are number of fixation points, right? And as there are number of fixation points, we call it like that person has a difficulty in recognizing or understanding that text. And also you also observe here there are multiple back and forth movement. Now these two are the features which are responsible or for which features that help us understand what is the language expertise of that person. At the same time, here, this person is very fluent in Hindi. That's why he is able to read it in one go, and there are less number of fixations. So, likewise, the reading studies were the first one where people were started using eye tracking. But can we just use the eye tracking to understand character recognition? So, as I move forward, like the second generation was the handwritten characters. So we decided to design our first experiment by taking inspiration from the handwritten characters. So you can see there are samples from the handwritten characters where you can observe that most of the variation is occurring in the body of that character. What I mean to say, if we divide the character into different sections, and then we can see that a particular section is getting modified. So what we did is we just divide the character in different sections and then scale that or distorted that section, and then perform like an eye tracking study wherein I'm showing the participant with the distorted letters and asking their response how at what level they can identify that particular character. And then we got to know about various distortions level that they can tolerate. Now these distortions and at the same time we also got collected their eye movement data and created a heat map of those eye fixations. Now if we look at these characters, look at their eye movement patterns, we observe that their eye focus is getting shifted to the different regions as this distortion level is decreasing. So it gives us some sense that okay, eye fixation could be a valid measure to understand strategies used for distorted character identification. But these distortions are created by us. So what if we want to extend this idea to the normal or real life examples of those characters? So what we did is we just try to because this all of this are the qualitative observations, like how and how many fixations there were, how their gaze pattern change over the period of time as the distortion level is changing. Now we wanted to understand their strategies for ornamental characters, which is like a special case of characters. So what we did, we created our own data set because there wasn't any calligraphic character data set available. So we divided or isolated each character from the word and did all the pre-processing to come up with final data set, which is like shown here, which is a data set comprised of ornamental characters. Now, experiment, then we decided to show these characters to the participant and then decided to design an eye tracking experiment. So here in general, when we talk about eye tracking experiment, the general eye tracking experiment protocol is you show a character or any stimulus followed by fixation cross at the center and then the next stimulus. But the problem with this is like when you show a stimulus at the center and also asking a participant to focus on the center, then that fixation on the earlier fixation cross may interfere when actual stimulus is presented. What I mean to say is you are focusing on this cross and then immediately the next character is appearing on the screen. You are actually, again, your focus is also on that. The initial fixation will not be that informative. So we propose to use this location of fixation cross, which is actually away from their actual stimulus size, so that when you start exploring the character, the initial fixations were away from the image and the actual information is recorded properly. So this is the final experimental protocol that we come up with, where we are showing this character for unlimited time, and it's a free viewing experiments wherein the participant was given freedom to come up with the decision on the character irrespective of time, and then read the character aloud so that we get to know whether the recognition was correct or not. Now here, I want to ask you one question. What is the letter that you see in the screen? What is this letter? You can write in the chat. We are presented with such kind of characters to the participants. So what do you think what this letter is? Yes, so I got the answer like P, D. Okay, now the context. It's a Hindi letter, not the English letter. Correct. So yeah, I got a nice response that it is letter G. So now when you are actually identifying this letter, you're again focusing on different regions of that image. So the person who said this P, their focus was on this portion, which is shown in the topmost gaze plot, and the person who are showing it as a girl, they actually focused on the uh, features which are actually relevant to the character. Now, based on your eye gazes, we created like map called as a fixation map, which actually summarizes the visual fixation information over multiple subjects viewing the same image. And then based on that, you can clearly say, okay, when the person is not identifying this character correctly, his focus is somewhere in the irrelevant portion of that character. Irrelevant as in the regions which are not contributing to the character identity. Whereas the people who are coming up with the exact correct answer, their focus was on the character regions which are actually contributing to the identity of that character. This is actually the letter G. Now, next question, what is this letter? Now, if I ask you this, what this letter is, you can say there are like, okay. So I got the answer like this is letter D or this is letter R. Now, um, we got the same different responses for the single letter. So how is this possible? Because the region you are actually focusing on this character decides which letter you are identifying. So the people who say that this is they might have focused on the top and bottom portions, whereas the people who are saying it as R has focused on this too, the topmost portion. So now, if you ask me which is this correct letter, I would say R is the correct answer because these letters or the database has been created by segmenting out each letter from the word image. So I was having the complete context while creating the data sets. Otherwise, if a normal person says is the and the, so I would say both of these are correct, but I am having the ground truth annotations available with this character. So then it is very clear that humans explanation can be derived from the eye tracking, and these maps gives us very informative, like the recognition involves information processing through different uh, regions through, and that can be analyzed through selective focus on that image regions. So now what about neural networks? How do we get the explanation from the neural network? Because we need an explanation which is similar, which is comparable to the explanation that are given by humans. So then it comes to the idea of explainable AI. I'm not sure how many of you are coming across this idea of explainable AI, but where we actually try to understand how deep networks take a decision in favor of a particular class in terms of a classification. So here, in most of the cases, deep nets are being used as a black box, where you fed the data to the deep net, they process the data and come up with some results. What is happening inside, we don't really know. which limits its application in the security system, surveillance, or medical applications. So, in order to deal with that, there are many techniques that are developed to come up with a probable or possible reasoning that are understandable by humans. So, we try to explore this black box or unbox it by understanding the internal operation to some extent. So these are the different techniques that have been proposed. Among those techniques, we also propose our own technique called as modified GradCam, that is gradient weighted class activation mapping technique. Now, these techniques help us understand where in the image a person is focusing. I'm not going to much details of this technique. I will just go to the results of this. So what is happening here, we are giving the input image to the model and then trying to look at the focus of that network, just like we saw the focus of humans. The fixation map is a human focus, and there are different techniques, and this is the technique that I propose, like modified guide gap. So on both on the qualitative and quantitative comparison, you can see that our technique is doing better compared to the previous techniques. So here, the idea is to identify which regions of the image has contributed more uh, for the network to take a decision in favor of a particular class. So if we give this image of row, if the network is focusing on this region, and then it is calling it as row, which is similar to that of humans, right? And we also compared this with very known technique called as GradCam, because ours is just a modification of the GradCam. So what we observe here is for the sample, like let's say 64, or for a batch of few samples, the GradCam is actually unable to generate any explanation for few images, whereas our technique is actually generating the explanation. So it means like through qualitative observations, we observe that, okay, our technique is doing better, but we also proved through analytically that probability of getting positive activation for our method is greater than the GradCam. So all these details are already there in my paper, which is published in IEEE Transactions in AI. So you can just go through it and let me know your thoughts. But now based on these two explanations, now let's try to compare the explanation of humans and the explanation generated by the technique that we proposed. So here, if we give input image to both of these, like input image to humans and same input image to the network, we see there is interesting observation that in case of a correctly classified samples, both humans and machines are focusing on a similar regions. Whereas when there is an incorrect classification, the focus is on different regions. So does it mean like it gives you a motivation like what if we make a model to focus on similar regions that are fixated by humans? For that, we come up with a model called as visual explanation guided attention model. So wherein we propose that we are taking the fixation map during the training of that model and try to make model focus on similar region as that of humans. And what we observe here, this is an input image, which is letter H. Initial model focus was somewhere here. Now, this is like the character image has been, or the heat map is overlaid on a character image. So this model is unable to identify this as H because his focus was somewhere at somewhere different locations. But whereas human focus was very accurate, and then when we did the training through this model, then as a result, we observe that the model is also focused as sort of regions of human. And now the letter like 0.6, 0.006, and zero point 0.7 are actually giving you the confidence corresponding to the true class, which is like this, if we look at the neural network architecture, it is just the final layer of your softmax, which is the maximum value, which decides the class label. And then these are some of the results that we clearly observe that incorrectly classified samples were getting correctly classified through our approach. And we also observe there is an improvement in the model accuracy. Here you can observe here like the red letters, the confidence in red is like less confidence corresponding to true class, whereas through our model, which is this column, column 4, it clearly shows there is improvement. Now, we observe like when we make a model to focus on similar regions as that of humans, we see there is improvement in accuracy. But what happens to the characters which are actually correctly classified? Are they remaining correctly classified or not? So we also saw that there is an improvement in their confidence corresponding to the true class. So the correctly classified samples are also remain as a correctly classified. They are not getting incorrectly classified. So then supervision using fixation map actually improving the performance of the classification. And also the interesting thing about this model is you are actually aware what is happening in the model. You are not randomly just doing the engineering approaches now, because the field of AI, what they are doing these days, you apply all sort of techniques to the neural networks, and then this, if some techniques works, then you just report it. Okay, we are coming up with this novel architecture and this is working so fast, because we actually have a very um, sophisticated hardwares which can do this computational task very easily. We don't really care about computational load, but we hardly know about what is happening inside it. But this model, you know what exactly is happening inside it. So it can find applications in medical or surveillance domain or security domain. Now, in some cases, we are not really, uh, we don't really want the explanation. We generally want like accuracy. And this is one more point where I'm trying to say like what like we can't really have fixation map for all the characters. So what happens when we just show the model or test the model with a sample which are not from the same distribution? Like what is the generalizability across different samples? So we tested this by showing a data set which is different from the data set which is used for training, and we observe that our model is actually doing better. But as I mentioned, we don't really need like an explanation for character recognition. Unless it is a medical application, we don't really care that much about what is happening inside the model. So can we just use the state-of-the-art architecture and use this eye fixation maps to train the model? So this is one of the model which is we call it a deeply supervised model, wherein what we are doing is we are trying to use the fixation maps to train the intermediate layers of the model, and then formulating it as a optimization problem with two losses, which cross entropy and um, mean square error. Uh, what we observe here is like when we make a model, look at the similar regions, we actually getting improved accuracy. Now, this work is also published in IGDAR conference, which is document recognition and analysis. Um, Now, in both of these models, the hypothesis is models should focus on the regions which are fixated by humans. But what if I say, can you identify this character? Can anyone say like which is this character? I'm not very sure. What about this? Yeah, thank you for your responses. This is so when I show the second image, the chances of getting it correctly classified is more, right? So this is the character K in the Devanagari script. So what humans are doing is we actually need both foveal and parafoveal regions for correct recognition. What I mean to say is like you have a focus point. At the same time, some information is actually there in your parafoveal area. that parafoveal information is your eye tracker is unable to capture. And that's why we the recognition is not correct in most of the cases. So if we analyze this data again carefully about this previous models, what I observe is there are some samples which are getting misclassified. For example, here the letter E, wherein human focus is on this center point, but we know that E will make more sense if we have those lines at the top and the bottom. So now this information is actually not been recorded, even though humans are using it, similar to that of Go also. So now we are having a problem like eye tracker is actually unable to capture the parafocal regions. So what we can do about it? So then we proposed a model based on the collaboration between human and machine. Now, if we look at the human brain, there are two different pathways. One is where and one is what pathways. So in the ventral stream, you can identify like as the information is processing from V1 to IT, we can have a similar analogy in convolution neural networks, which is like this ventral pathways are called as what pathways and the dorsal is where pathways. So if we look at the CNN architecture, we can also say like the spatial extent is called as where, and each channel could be a what, kind of give us a sort of similar analogy as that of dorsal and ventral pathways. So now using these two ideas, what we decided to do is we decided to take the focus point from the visual attention map, which is nothing but a fixation map, at the same time ask the network to take care of the parafoveal regions. Now, this network attention map has been decided, like you can see here, and then we say like this NM takes care of the parafoveal regions. And then in order to decide that, we did a very like in general, people do the ablation experiments. We just did many experiments. What should be the methodology that we should be using, whether we should use average pooling or max pooling or concatenation of average or max, and whether this multiplication of the network attention map and human map should be like dot product, or it should be any element-wise summation or element-wise multiplication. At the same time, how should we combine? What is the significance of using a fixation map? Can we just use the network attention map just to come up with the idea of just to use a spatial attention map? So now what we observe here is like, what if we don't really use the fixation map? So what is happening here? This is an input image. This is a baseline model where we are not doing anything, it just without fixation map being used for the training. And then third is like a fixation map. When we don't really use the fixation map, the spatial attention map is doing quite a good job. But at the same time, when we do a collaborative attention model for the topmost row that I'm showing you, here you see the focus points are more focused to the um, regions which are actually relieving the identity of that character. At the same time, we have this letter the, which makes the case more stronger. And we also observe there is an improvement in the accuracy of the performance. So if we compare these models together, we can see like the collaborative attention model is doing their job pretty well, as they are taking care of both attention map, like human attention, at the same time network attention in terms of foveal and parafoveal regions. And we also compare our model against different models which uses eye tracking methods in some sense, not exactly for the characters, but for the radiology images. We just use those architectures just to compare our results with that. And if you look at this generalization ability, like or just using the handwritten and transform characters to test the model, which were not being used while we train the model, we also observe that when we are actually using the fixation maps, the model is generalizing pretty well. But the question is still remaining is like human recognition performance in all these data sets is nearly 100%. So we are still not at the level of human performance. So there is on the first part, the concluding remark that I have is like recognition is actually a multimodal approach. So here you can see the person is trying to identify the character which is displayed on the screen. At the same time, what she is trying to do is she is trying to write it. So it's just not the visual thing, visual recognition, but also your motor cortex is also involved in the recognition. Now, when we train a model, we are just using images as an input, but we never thought about training a model with a multimodal approach. Now, this is the something new that we need to use a different, like the if we use the engineering based approaches by augmenting them with the findings from such cognitive experimentation, we can try to narrow down the gap between human and artificial intelligence. So now this is the future of this work, that if we want to take this further, one can design many different sort of cognitive experiments to come up with a better model. And also there was a need, like this work that I did, it's like my PhD work, which I did at IIT Delhi. So now this work ended like we need to understand more cognitive processing that are happening in your human brain. And that lead me to take up a position in brain and cognitive sciences to understand how brain make a sense of the world. So I'm quickly gonna share some slides on this project. It's called as a Project Prakash, which goes like illuminating lives and illuminating sciences. So this is like 1 in every 100 Indian is blind. And the incidence of childhood blindness in India is 3 times than that of in the US. So now why this is the situation? What are the consequences of childhood blindness? So over the 40% of the childhood blindness in India is actually treatable, but unfortunately because of uh, like unprivileged population, or people cannot really afford the medical care uh, for treating those blindness. That's why most of the children are remaining blind, even though this blindness can be treatable. As a result, because of this blindness, the lifespan is getting reduced, and also the education and employment prospects of such children are very less, which is actually the situation which is devastating for anyone to believe. Like, why are in our country we say like India is getting developed? I mean, it's a developing country, but we are making progress in a developed country, but we can't really provide the medical care to these children. Now, there is actually a need to provide treatment to such curably blind children, which is a humanitarian aspect. But this also gives us some scientific opportunity, like called as a window into early visual development. Because as the children are born blind, they are blind for the several years of their lives, and they might have missed some of the critical period where the actual development of certain abilities should be should happen or should occur, but they might have missed those periods. So what are the consequences of such things? This is like a two-way thing. One is a scientific quest, at the same time there is a humanitarian need which merges together, and then it gives rise to the Project Prakash. Project Prakash is being founded by Professor Pawan Sinha, who is my current PI, and I am working with this project like since I joined postdoc, and also before joining postdoc, I was working with this project, which is both the scientific and service-based project. So the Project Prakash has streamed. I'm really fortunate that Professor Sinha has given me a chance to be a part of this project. So then this project has three parts. One is outreach, treatment, and then research. So in outreach section, so currently our headquarters is based in Delhi, and this Project Prakash is actually covering the northern part of India at present. But the plan is to expand scale all over India and at the same time all over the world. So now that outreach team in Project Prakash go to different villages and try to identify the children who are having treatable blindness. And they are called into the Shroff Charity Eye Hospital, which is situated in New Delhi. They get the treatment. At the same time, if they are willing to participate in the studies, which gives us an opportunity to understand how brain learns. They are getting treatment to the world-class surgeons in Shroff Charity Eye Hospital, and then it gives you a like of an opportunity for us to understand how vision develops after sight onset. In order to understand how vision develops, the first question was whether actually the vision develops in such children. So Project Prakash, I would suggest the participants just to go to the TED Talks, which has been given by Professor Pawan Sinha, which will actually, it's a better way how he explains it. And we found that actually vision develops after the sight onset. So in that project, I was like involved in multiple sub projects, which are under Project Prakash, like EEG, biological motion detection using eye tracking, at the same time communication tasks, where we are seeing like what are the neural changes that are happening, at the same time what are the communication skills are getting improved. So this project has given me like multidisciplinary sense and combining different the knowledge from different fields, like from psychology uh, to the cognitive sciences, the linguistics, and AI, of course. So now this project has also had a very broader impact in understanding this different neurological disorders as well. I wouldn't be talking much about on this, but I will just quickly get you through the project that I am currently working in, Project Prakash, and at MIT. Is like development of face perception in congenitally blind children. We call it as a Prakash children. So now I'm not very sure about how much background you everyone in the audience has in terms of an EEG signal. So electroencephalography. So in this image, you can see me. So interestingly, my work has been like our work has been published in one of the Geo magazines, and this is a photo from that magazine. So here you can see like we asked the participant to wear this cap. I hope my cursor is visible. And these are the electrodes that have been placed. So electroencephalography actually records the electrical activity that is happening inside your brain. So you know the neurons get fires, and if there are certain neurons which are firing at the same time, that gives some sort of a signal at the surface level, and that signal has been captured using EEG. So um, and EEG is like it has very much high temporal resolution. So we can see at what time what is happening inside the brain. So now we ask the child to wear this cap, and then we prepare a subject and show them the stimulus and record their brain signals. So here the particularly I was talking about face recognition or face identification expertise. So you know there is a marker called as N170 which shows a typical which is responsible for face, giving us a sense like whether a particular person is expert or is able to identify faces or not. But whether this ability of recognizing faces develops through develops over a period of time, or is it like a person is born with this ability. So these are the two recent work which is says like N170 shows typical adult like profile as early as 5 years of this age, suggesting that at the age of 5 years, you get enough visual experience that help us to identify a particular stimulus as a face. Now, this there is like they also suggest that if a person is born blind and had gone cataract surgeries between 2 months to 14 years of age, the ability to interpret the particular visual stimulus as a face uh, is is actually dependent on the early visual development. So these children won't be able to identify this as a face or non-face. So now, or whether their neural changes are accompanied to comment on this. So then we have two or three research questions, like whether this ability to recognize faces is innate, that is um, by birth it is there, or it develops the experience, or how much visual experience is needed if it is developing over the time, and also is this um, compromised, or whether it's similar to that of healthy controls. So we took up this challenge and then decided to design our own experiments, wherein I'm showing this object images and face images to the participant. So this is an experimental protocol where we are showing the participant with different images of faces and objects. And we took this recording at 3 time point, like pre-operation, post-operation, 1 month after the operation, 60 days after the operation, after a year. Like this, we longitudinally taking the recordings of these children. And then I'm just showing the results of the ERP analysis. So ERP is event-related potentials. So what is happening here is, let me just try to get a pen. So generally your EEG signal is something like this, which is a continuous signal. Now, as I'm saying, like we are showing them images at different time points. So let's say there at this time point, face image appears. So this is, we just mark this image as like this EEG signal with a marker called as S1. And let's say after some time, as these images are presented at random, so it is called as S2. Sorry, pardon me about my, because I'm using the mouse. So likewise, we have multiple, like 100 trials for faces and 100 trials for objects. Now we cut this signal from here to, let's say, here with, we want to take a signal of, let's say, 900 or 1000 milliseconds. Okay. Now these 1000 millisecond segments are collected over the whole EEG EEG signal, and then we cut it into different segments. And now these segments are averaged together to give you an averaged ERP response, which is we call it as a ERP. Now here we see the first peak, which generally occurs around 100 milliseconds. Now in your brain, the information is passing from vent over the ventral stream, um, and then it's like the initial processing of the stimulus is happening around 100 milliseconds. And now when you go for the N170, what I mean to say is once the stimulus is presented, after 170 milliseconds, you see a negative dip. Now this is a kind of a marker for face identification. And as we go further, then there are more cognitive processing that are happening in your brain. So you can infer something from this. So we are mostly interested in N170, which is the negative peak that is happening here. Now here I'm showing the results of one of the participant from the Prakash. So here we see this pre-operation. So here VA is the visual acuity, which is the sharpness of your vision. In normal adults, it's 20 by 20. When you say like, what is your vision? Is it corrected through glasses or not? We generally compare it to like 20 by 20 here. Pre-operatively, their visual acuity is 20 or 1016. They can, they hardly have a light perception, but they can't really say what the image is, and which is also very clear from their neural responses. Here you can see that there is no distinction between face and object. Even after the operation, there is no distinction. But after like a month, you can see there is a distinction between this N172 component. So we also observe it's like rather than looking at this P8 electrodes, which is this electrode, the right hemisphere is actually dominant in face perception. So why not to combine all the electrodes which are the right portion, like P4, P08, P04. And if we combine this, we can see there is a distinction between face and object, neural marker. And then if we compare their amplitude and the latency, we can clearly see like whether this difference, like the difference that I showed, is this a statistical significant or not, or it's the same. So we observe that there is a statistical difference between the phase and object amplitude. Not phase and object amplitude, is the amplitude corresponding to the phase presentation and object presentation. And it is getting more stronger. So here is the one star is like less p-value less than 0.05, and here the p-value is less than 0.01. So we can clearly see that this, so I'm just showing the results of 2 patients, but I have a result for like 10 patients from the Prakash cohort at different time point. And when we compare this to that of the control or healthy participant, we can see, now this is the healthy participant, we can see there is actually a distinction between face and object, and here also the age match participant is also showing the same distinction. So this results actually gave us an idea like our brain is plastic enough to accommodate some changes or some abilities. So now the neural markers of face perception is actually emerging after a few weeks after the surgeries. It means that we are observing these changes, and it is dependent on the visual experience that is which are acquiring which is acquired for the face categorization skill. And this finding is very stronger finding as compared to the earlier claims, like face recognition actually can be acquired late in life. So what I mean to say is your neural network of your brain is getting reorganized in some sense and giving you a better neural response over the period of time. So now these findings have implication for understanding the brain plasticity as well as object representation in our brain. So now there are multiple projects that this is one of the snippet that I shared in the project Prakash, but we look at their visuomotor coordination and multimodal interactions. And other than that, we also that project has also helped us to define the regimes of training, how we should train a neural network similar to that of, is it helpful like the babies are born with a blur vision. What if we also train our model with initially the blur images and later on with the color images? Do we see the improvement in the performance of the model? So all these sort of questions are being addressed by Project Prakash, and this children have actually given us an opportunity to work with this wonderful project. And I would conclude by acknowledging the people who are actually co- um, contributed this project. So it's like I would like to thank my PhD supervisors, Professor Tapan Gandhi and Shantanu Chaudhary, and currently my PI here, Professor Pavan Sinha, and all the collaborators that I had from India as well as from the US. So thank you so much. I'm happy to take any questions.
So I think we can open the forum for question answers. So if anybody has some questions, they may ask. Um, I think I should start with the questions because I have 2 questions. So
Okay.
one is, when you showed some letters to us, it was
Mm-hmm.
not decisive, or we couldn't at a glance decide what the letter was. So does the gaze time play an important role while recognizing these letters? Like if you let the letter stay for a longer duration of time, or we stare at it for a longer duration of time, maybe our brain intuitively can conclude that this might be the letter. So in your experiments, did you have a specific gaze time, or it was random?
No, yeah, thank you for this question. So this was a free weaving experiments wherein we gave a participant as much time as they want. There wasn't any time limit on it. And the idea was exactly the same. Like these characters are a bit complicated characters. And we kept it complicated because we wanted the person to inspect to the each and every regions in an image and then come up with a accurate answer that this letter is R, this letter is G. What if I just show that letter like a printed sort or which is not that complicated? You actually need a second or less than that just to see it and then say, okay, this letter is Ka. But we wanted to get a more informative fixation maps. That's why these letters were complicated. And that's why we gave them as much time as they want. There wasn't much time limitations, but at the end, while when you spend more time, you are actually taking out the information from different regions and then combining it. That's why you are taking more time to identify. So definitely the time is a crucial factor, like time that has been spent on each character to come up with a decision. That's what we wanted to have in our
So I take
feed.
privilege of asking you another question. That does intuitive capacity of individual's brain also play an important part in recognizing the letters or patterns, because since our brain, it favors the continuity in patterns. So does
Mm,
this individual's capacity, intuitive capacity, plays an important role here?
yeah, it depends on the experience that you had with this. So now each person is having a different experience with this letter. They might have like, so all the participants which participate in this study were like fluent in Hindi or Marathi or whichever language wherein the Devanagari script is being used. So each person has a different way of interpreting that particular character or focusing on it, and they are focusing on different regions. But ultimately, yeah, so the experience with that letter plays an important role, I must say, in identifying or giving us me the recognition result.
Thank you so much for explanation. Uh, if there are any other questions, we can proceed. Participants? Okay, so I think you have given enough food for thought to everybody.
Yep.
Uh, so on behalf of the organizing committee and all the participants of this IFDP, International FDP, I
I
would
think
like to
somebody
express,
raised the
yes,
hand, Manish.
somebody has raised the hand, I guess. Participants who want to ask question can write your question in the chat box, please.
Oh, I thought they might unmute themselves.
So I guess it was by mistake. Yes, so on behalf of the organizing committee and all the participants of this international FDP, I would like to express my heartfelt gratitude to Dr. Chetan Rayakar for his invaluable contribution as an expert in this FDP. And your research in the artificial intelligence and neuroscience has provided with us with the profound insights and practical knowledge, which will undoubtedly enrich our teaching and research endeavors. So I once again thank you so much, Dr. Rayakar, for your valuable time and gracing the occasion.
Thank you. But I think that person wants to unmute and ask the question, I
Yes,
guess.
thank you so much, sir. It was
Yeah.
not mistakenly. Yes, thank you so much for the presentation you have given, sir, and introducing us with Project Prakash.
Mhm.
And one more thing, sir, that it beyond the humanitarian impact it's have, apart from that, it's open the challenges and the fundamentals behind how a human brain works or capable of seeing the visual world. Thank you so much, sir.
Yep. Thank you so much for this wonderful comment. And I would definitely recommend you to just go to the TED Talks. It will actually give you a sense actually what's happening in Project Prakash. And this is really a wonderful opportunity that I got to work in Project Prakash at MIT and in India. And it's really nice to like sitting in the West, like I'm in Boston currently. So being in the US side, I'm able to serve my country in some sense. So it's really a great opportunity for me also to work. And I would definitely love to thank the organizers of this workshop. And I hope that some of the information was like useful for the participant. And I look forward to meeting my alma mater soon, sometime in person. So thank you so much, everyone.
We will surely love to have you here, and we are waiting for you to come to India and meet us. Thank you so
Well,
much.
thank you. Thank you so much.
So I think today's sessions have ended, and I request all the participants to join the session tomorrow on time. Thank you, participants.
Thank you.